Great. Thanks, Matt. That's very nice. And thank you for inviting me. It's a real honor to be here and to get to give a talk at this amazing celebration. I'll do this with my left hand. All right, so I'll just get straight into it. The question is, I'm posing is, do animals have imagination? And I'll try and answer that question and then tell you why I think it's interesting. But we start with the hippocampus, right, which everybody here knows about. It's the original organ of memory. It's well known since patient HM, who was studied for many years not far from here, just a few blocks down the road, by Sue Cork. And the removal of his hippocampus left him with an interrogated amnesia, so he was unable to form new memories. And so we all know about it. Even the logo here shows the hippocampus, so we all are interested in that. What kind of memory? That was one of the questions. So if I was to ask one of you, what did you have for breakfast this morning? You didn't know I was going to ask you that question, but you can answer that question by going back in your memory and reimagining yourself at your breakfast table or out there where you got a croissant or something, and reliving that experience. And so it's that kind of memory that the hippocampus seems to serve. So we've known about this for a very long time, obviously, in the field. But a very interesting twist on this was done in a study of patients with hippocampal damage, but asking a different kind of question, which is getting them to imagine themselves in scenarios that they've not been in. So in this study by Demis Hassabis and colleagues, he would ask them a question. Can you imagine that you're lying on a beach in a beautiful tropical bay? And so a normal patient without damage can spin a nice long narrative story. You know, I'm lying there and it's really hot. I can feel everything, the sand underneath my feet, and I can see someone passing by in a boat and I wave at them. But a patient with hippocampal damage isn't able to do that. So if you read what they say, it's a bit hard for me to read it over here, but a typical response would be, you know, I can basically see some things, but as for seeing, I can't really see anything apart from just sky. That's what one of the patients said. I can hear the sound of seagulls, but then the experimenter asks the person, are you really seeing anything? And they say, well, not really, just the color blue. And so, are you seeing this in your mind's eye? No, I can hear something, but it's kind of like floating. So there seems to be, at the core of this hippocampal function, a kind of reimagining function that may be also how that kind of memory works. So we study this in an animal model in rats, and this is the hippocampus in a rat. It's a much larger proportion of the rat brain. And this is the classic cross-section drawing by Cajal, but you can see the output neurons, the cells of the hippocampus that relay information out to the rest of the brain. And what we do, the technique that I learned in Matt's lab, is we drop these electrodes into the brain, so we record from cells in the hippocampus while the rats are running around and having experiences. So they're fully awake and they're behaving and hopefully forming memories and retrieving them. So what do you find? The classic finding in a rat, I'll just explain this slowly, is a place cell. So what you're looking at is you're looking above an arena that's about 2 meters square. And in black, you can see a search path of a rat just running around looking for food for about 20 minutes. You can see it just runs all over this environment. And what you can see in red is everywhere when one hippocampal neuron fired a spike, the action potential, which is how neurons communicate with other neurons in the brain. And you can see, I think, very clearly that this one neuron has a preference for a particular point in space. One of the earliest results that sort of showed that across many different neurons you get a kind of map of space was this result from Matt Wilson's postdoc, actually, And this is the paper that actually inspired me. After I read this paper, I wanted to join Matt's lab. So this is what made me want to join. And you can see here that in what you're looking at here is depictions of the responses of many cells. Each box that you're looking at is a depiction of the arena that the rat is running around in. And the heat map shows you where one of those cells preferred to be active. But you can see these are all recorded at the same time. So you can see that across these cells, there's really a map of space. So this is a pretty amazing discovery about what happens in the hippocampus in rats. It was half of the Nobel Prize in 2014. But it leaves a huge question, which is, what's this got to do with memory? When a rat is in one of these places, a number of cells will fire. But what does that tell you about what the rat is thinking about, or what he's remembering, or what he's planning to do? Not much. So a long series of experiments. I was trying to work towards getting an answer to this question. I did a few experiments in Matt's lab, and then lots of other people in the field were doing similar experiments. And I'm going to skip over all of that and get to an experiment I was able to finally do in my own lab, where we got a first pass at an answer to this question. So we have a rat trained, again, in a square arena, about 2 meters across. And here the animal is doing a spatial memory task. So these are little wells where liquid chocolate can bubble up that the animal likes to eat. And the task here is he has to explore around the environment to find chocolate in a place that's unpredictable. And then, on the next trial, he can go back to a predictable location where the chocolate will come up. And then he has to go around and look for another random location. And then he can go back to the predictable location. So it's constantly toggling between random foraging and memory-directed behavior. And we can tell that he can remember it because he's much faster getting to the food when it's a predictable memory event than otherwise. We increased the number of electrodes that we use. So this is my version of the Matt Wilson figure. And you can see we have lots of cells, but again, place fields, except for the handful of cells that fire throughout the environment, which we then can exclude independently as interneurons. But for all the other cells, you have this very clear place field response. And so these cells are all recorded simultaneously, but across all of them you have a map. But what are we going to do with it, right? So we have to somehow extract from this population some message. So this is how we do it. Just to summarize, actually a lot of math, but if we have 4 cells that have overlapping fields, and they all fire some spikes, we can say we have a strong belief that the hippocampus is telling us that the animal is in this location. So with that kind of information, we can construct these movies of what's going on. So now you're just going to watch what the hippocampus is doing in real time as the animal is running around in this task. And so you can see a depiction of the animal's position and its direction. And then underneath that, you can see this flickering. That's the decoded representation of the location from the place cells. That's the overlap of all the place cells that happen to be firing at any moment, right? And you can see it tracks the animal's position pretty well. But the interesting thing is what happens when he stops. And Matt alluded to this in terms of awake replay. So the next movie shows a number of events that happened while the animal paused immediately after that exploration. And they're happening much more quickly than the animal's behavior. So this movie is now slowed down by a factor of 20. But what you can see is that the representation of the animal's location actually moves smoothly away from its current location, directly towards that remembered home location. And that, we think is an interesting model of the animal remembering and planning what it's going to do. So across a number of these events collapsed in time, we found that many of them will start in different locations and proceed towards the remembered location and could actually predict what the animal was going to do next. So I'll skip over some of the statistics by which we proved it, but for example, just comparing the replays with either the past path or the future path the animals are going to take. There's a bias towards predicting the future path. But I want to draw your attention to a little thing that I didn't tell you, which is every day we actually play a trick. We move the location of this home. So he has to actually learn a whole new home location every day. Why is that interesting? Because the random locations also don't repeat within the day. So if you think about it, what's happening is, on every single trial, there's a new combination of start and end location. So every single time the animal arrives at one of these random locations, he has to make a new movement that, in terms, I mean, the space is very familiar, but in terms of start and end location, it's a novel combination. So I'm going to say, maybe this requires a kind of imagination. So it's not strictly memory, it's the creation of a new kind of representation which corresponds to imagining what the animal is going to do next. So this is a movie that combines the behavioral timescale with this replay timescale, and you can sort of see a whole sequence of things happening. So in this case, the animal is moving around the environment. He's looking for this random well, and then when he finds it, now we slow down time, and you see a number of these events occur in succession that depict the home well, and another location that he seems to be interested in too. So there are a number of these replays, and then we go back to a behavioral timescale, see what he does next. But that's the first time he's actually had to move in that combination of start and end point, right? But the movie continues, actually. It turns out that he was interested in that little corner, and he has a couple of replays that go in that direction, and then he actually goes there. So it's constantly predicting what the animal's going to do next. I can give you a summary of the summary. We found these interesting predictive dynamic representations that we think could be a model of the animal imagining his future. Now, I'm now at Berkeley. Berkeley is famous for many things, but it's famous in the early part of the 20th century for this fellow, Tolman, who did a number of, he was very concerned with trying to show that rats were more intelligent than people were giving them credit for. And so he came up with a whole number of different kinds of mazes to try and show that the cognitive abilities of rats could not be accounted for by very simple reinforcement learning rules, but had to involve some kind of internal model of the world. Inspired by that, my postdoc, John Widlowski, developed a more challenging version of the task that I already just told you about. In this version, again, there's a square maze. Again, there are these locations that have to be either foraged for or remembered. But John put in these barriers. So if you look here, there are these, he calls them jail bar barriers. So they're actually, the animal can see through them. He can poke his nose through them and smell through them. The only thing he can't do is walk through them. Right? And then the idea was that we would place these barriers in a number of different configurations, more or less randomly, in random permutations every day. And so the animal is constantly being exposed to different permutations as we, so when we change the session, we change where the home well is, but we also change all the barriers, right? So it's actually a pretty difficult task. I'm not sure I'd be able to do it. And you can imagine changing all the walls in a room, and they go through 100 of these different configurations. So just summarize this. The behavior shows that they're able to do it. They're able to move more quickly to a remembered well in this environment than to when they're randomly foraging, which makes a lot of sense. But let's start with what did the place cells do. So this is the third figure that you've seen. It's the same kind of thing. It's a bunch of simultaneously recorded place fields of these cells. But now they're grouped into threes. And the three is the same cell recorded in 3 different sessions with 3 different barrier configurations, actually in the same day. So I think it was 12 p.m., 3 p.m., and 6 p.m. In the same day. And what you can see is that the representation of space is pretty stable, which was actually a surprise. We might have expected that these barrier changes would lead to a whole-scale, what's called remapping of the environment, but they didn't. Some cells show some small local changes, but many of the cells just consistently do the same thing, despite all of these changes. But what's going on inside the hippocampus in terms of the dynamic activity? Here's an example of what you can see. Now, it's not a movie. I've collapsed it, so the color tells you the passage of time. But here's the rat. He's completely stationary, just like before. He's looking off towards one wall here in the direction of this line. But a sequence is emitted across the cells that just nicely goes around the barrier. And this is a barrier configuration that he's only just had a little bit of experience with. So somehow the system adapts and is able to predict the future around these different barrier configurations. Here are all of these replays. So these are all activity patterns that occurred while the animal was stationary. But you can see so it was 3 p.m., 6 p.m., and 9:30 p.m. Okay. But we changed the barriers, but you can see that the replays really beautifully capture this structure. So even the animal's not looking at the barriers when this happens, and it doesn't correspond to what he's just recently done. It's not some kind of simple buffer of experience. He's really sitting there and imagining his way around these just recently learned barrier configurations. And you can see this very different structure of replays depending on what's allowed. And of course, because it's all in the animal's head, it didn't have to be like this. It could have been that the replays just go straight through the barriers because it's just virtual, right? But in fact, he's rapidly learning that structure. And so here's just some examples. And you can see that in many cases, the animal's just sitting there and the replay will go straight to the home location and predict what the animal has to do next. So rats can imagine spatial movements. So what? Why do we care? Well, I think for me, the big point is that you and I are using the same piece of machinery that the rat is. So let me back up. We know that our brains are much more complex than a rat's brain. And physically, there are certain parts of the brain, the frontal lobes, for example, which are far larger, much more developed, much more complex than in a rat. But the hippocampus is a very old structure in evolutionary terms, and it hasn't changed that much. And so that's evolution, folks. All your complex memories but have to be channeled through this old piece of machinery. So I'm sure that you're not only imagining spatial trajectories, but I think that these sequences and this kind of logic is going to be there also. And this is important because, to be serious for a second, there are many diseases of the hippocampus, and they affect memory, and we really need to find a way to address these problems. So the first step is to understand how it works in this animal model. And the good news is, in a sense, we're not that far off. So if you think about a downstream neuron in the entorhinal cortex is receiving this information from the hippocampus, it's sampling about 10,000 neurons. I mean, it's doing what we're doing. It's listening to the signals and trying to make sense of them from about 10,000. We're sampling about 1,000. If you include all of the silent cells. So we're getting pretty close. And at some point, we'll have a fairly complete description, I think, of all the kinds of activity patterns that are possible in the hippocampus. Step 2 is to figure out how to manipulate the activity. So we're already starting that in my lab, and there are several other labs that are interested in this too. And so the kind of questions which are clearly translational include, how can you suppress memories that you don't want to have? How can you enhance memories that maybe have been weakened in disease? But ultimately, how can we replace this entire piece of machinery with a prosthetic that does exactly the same thing? So I want to acknowledge my members of my lab and funding sources. But seeing as I'm here, the person I want to acknowledge, above all, this fellow. So it's true, I was here as a student, visiting student, and now I was here as a postdoc. I mean, being here was a transformational experience for me. And, you know, the entire environment was an incredible place to be educated. But also Matt in particular, who, you know, is kind of softly spoken, so you might not realize it, but he is a source of trenchant opinions and deep thinking, and he completely changed my opinion about a number of things. You know, I came in kind of fresh-faced, and he kind of sorted me out, and I've been kind of trading on his ideas and plans ever since, and that's him listening to what I'm saying. So thank you for listening, but I would like to give Matt a round of applause if he will join with me.
All right, thanks a lot, David. I think we have time for maybe one or two brief questions from the audience. We'll start up front.
Wonderful talk, Professor. I'm wondering if anything is known about the molecular basis which encodes the information for the engram in the place cells.
The short answer is we know quite a lot, and a lot of people are studying that. It's an interesting point because a lot of work traditionally has focused on trying to understand how molecular changes, particularly synapses, leads to behavioral changes. In a way, all this place cell stuff is interposed between those two, and it has to make sense. So one of the classic results was, or my other mentor, you know, was Richard Morris, was that you can block NMDA receptors and block a certain kind of synaptic plasticity and lead to memory problems. The funny thing is, you could do that in the place cells. The place fields wouldn't really change at all. So it turns out the replay does change. So that, you know, there is this necessity to link across levels, and it's not trivial. But the short answer is there's a lot of work, including by many people here, looking at molecular mechanisms for learning in the hippocampus.
So to keep this show going, I'll just ask everyone to save any of your questions for the coffee break that we have after a couple of talks. But let's give one more round of applause for David, please.